Hello. So welcome back to PN4032, and we are looking at lecture 8, and we're continuing on with tree growth, and on Friday's lecture we'll be going on to wood drying. Okay, so first of all, congratulations on completing your first MCQ. Results were a bit varied. There was a lot of people that had some very good results. Content wasn't very complicated. It will be a little bit more complicated in the following MCQs. So if you feel you struggled, or if you feel you got a few answers wrong, it might be time to kind of pull up the socks and start doing a bit more with regards participation in lectures and participation in lab activities. Okay, so same as every week, just looking at a bit of a recap on what we've seen so far. So we've talked about Ireland's history and wood. We've had a quick look at silviculture. You'll have to look at that a bit more in your own time when you have been doing so in labs and creating different posters based on a forestry stand close by to a school. Growth of forests, so how do trees grow, how do we maintain forests, how do we take care of forests, that's all part of silviculture. Scientific method, we had a quick look over that just to try and maintain our own applications of that in this module and in the future. Wood use, so norms and innovations, what can we do with them, what has been done with it. Growth of wood, and cellular structure of trees where we are now, so the distinction between xylem and phloem, we have that. The evolution of trachoids and angiosperms, so what they can turn into. So that was in our last lecture on Thursday, and accompanying that was a Google Forms, and there's very little engagement with Google Forms. I need you to complete that Google Forms if you can, and let me be able to get an understanding, or help me develop a better understanding as to what stage you're at as far as your knowledge around trachoids. So just having a quick look at this again. So this will be due for your next lab next week, as far as I can remember. So it's due for next week's labs. It's your application principle 2, so it's based on tree growth. So in your first application principle submission, you detailed your approach to growing a forest close to a nearby school. There's a secondary school just down the road from your forest. You've been tasked with creating an A3 sized infograph which explains the mechanism by which a tree grows. This is to be displayed at the entrance of the forest and designed to educate students in secondary school. This is directly related to the content covered in lectures on tree growth. The infograph must be submitted as a PDF. So I couldn't break this down anymore. It's fairly simple. It's nice and easily understood. So you're basically going out here now to make an A3 poster explaining how a tree actually grows. That can be taken in whatever way you like. If you're going to take a poster and design it and create it to show us how photosynthesis works, okay, very good. We haven't covered the topic of photosynthesis. It's been very vaguely touched on. We want to learn a bit more about the mechanism by which a tree grows. So this is more so about the phloem and xylem tissues and what's going on in the cambium layer. How is the tree actually growing? Can you explain it to people? Can you explain to them that the bark is being pushed out to the outside? Can you explain to them that the tree might not necessarily grow out as far as it grows up? What is actually going on? And try and do it in a nice, clear way. Being able to explain the parts of the inside of the tree could be a good part to add on to this poster. Okay, so prompt and question. If you were to insert a screw in a tree at 1.3 meters off the ground today, and in the next year the tree grows 500 mil, okay, what height would the screw be at a year from now? So would it be greater than 1.3, equal to 1.3, or less than 1.3? So that's something that we're kind of interested in looking at. So we've got primary and secondary growth in a tree. So this image here it looks like an ash bud. Alright, and if we're looking at it, we can see we've got kind of shoots coming from fresh buds that are kind of primary growth. So we're growing up more than we're growing out. Okay, but has that original branch really moved in position? Not really. Okay, so there's very little kind of primary growth pushing existing branches further up the tree or to a greater height, whereas more shoots and fresh growth will grow off the end of them. Now there is growth in primary growth where branches will push up, but the majority of growth will occur through shoots, such as you can see in the image here. As well as that, we then have our secondary growth where the tree will start to grow in width and will start to push things further out. So it will move. How much is it going to move? It's more than likely going to be very close or just slightly above the 1.3 mark, because it will grow up slightly, but the majority of our primary growth will come from our shoots, and then our secondary growth is where we increase in diameter as we're going around. So we're going to be having a look at that, and we're going to have a look at the cells that allow for this to occur now in this lecture. So, types of tree growth. We've got primary and we've got our secondary tree growth. So if we're looking at our primary, so if we're looking at our primary growth, we're talking about the upward motion of growth, and our secondary growth, we're talking about the increase in diameter. Okay, so this graphic is a nice little demonstration explaining how the growth will occur in the tree. Okay, so if we start here, we've got our first year of growth, basically starting from the pit and moving out around the tree. So our vertical height, or primary growth, it's almost like a next layer comes on top of the annual ring, and it's almost a conical structure with each individual year. Yeah, and we can see that as we go out through the knot structures as well. That can be seen when we do our rip cuts, and we go through knots in different directions. We can clearly see the kind of conical structure to them sometimes. But as we're growing, our primary structure is a conical shape, working on the existing year previous. Okay, and we've got our majority of our growth is occurring from that location. What's important to recognize is if you have a knot at a certain location, so say here in year 3, the knot would have originated as a shoot. It would have started to grow here at this location, you know. The oldest year we have here is a 60 year of wood growth. If we follow that annual ring down the circumference of the tree and to the top of that current knot that we're talking about, yes, it has increased in height if we were to look at where the knot originated from. That is more so to do with the secondary growth that's also occurring. So there is diameter increase here, but the original height of the first knot, or the first branch, is still more or less the same. It hasn't gained any huge amount of primary growth in regards to its location on the trunk of the tree. So there's minimal primary height for something that might be damaged on the surface, such as a nail being put in or someone damaging the bark. That height difference will not increase massively across the course of the tree's life. Okay, and then the secondary growth is our girth, or the diameter of the tree, which we understand grows, which we understand will increase based on the quality of growth that can occur in the year between our winter growth and our summer growth, creating an annual ring on the tree. Okay, so meristem. This is something that if you've done biology or ag science, you might have an idea of what these are. So meristem is a region of cells capable of division and growth in plants. Meristem are classified by their location in the plant as apical, located at root and shoot tips. So lateral in the vessels and core cambium, and intercalary at internodes or stem regions between the places at which leaves attach and leaf bases, especially of certain monocotyledons, example grasses. So apical meristems give rise to primary plant body and are responsible for the extension of the roots and shoots. Lateral meristems are known as secondary meristems because they are responsible for secondary growth or increase in stem girth and thickness. Meristem form anew from new cells in injured tissue and are responsible for wound healing. Unlike most animals, plants continue to grow throughout their entire lifespan because of unlimited division of meristem regions. Right, so if you read through that, there's actually a lot of information going on in that little small paragraph, right? So the main things we need to talk about there is we need to have a look at apical and lateral meristems. They're kind of the two things we're going to come across the most. So apical meristems are located at the root and the shoots of the tree, okay, and that's allowing for our primary growth to take place. So the meristem is where growth occurs, and it's where our cells are able to divide, and there's an unlimited amount of division that can occur to allow for the growth to keep continuing. So the tree will continue to grow throughout its entire lifespan. Apical ones are for vertical growth or primary growth, and then lateral is for the diameter of the tree or the secondary growth. Okay. Another interesting thing there, and if you think about it, it makes sense. So a tree will continue to grow, or a plant will continue to grow throughout its entire lifespan because of the unlimited division of meristem regions. Okay, so animals will reach their kind of max size at a certain age of maturity. The meristems will have ceased then, and growth will slow down and eventually stop. Whereas a tree or a plant will continue to grow throughout its entire life cycle. It can, in some ways, begin to slow its kind of primary and secondary growth, coming to its last couple of years in growth, especially when you're looking at the likes of forestry. However, there is still growth taking place, so the meristem is unlimited in its accountability for growth. The other thing we want to have a look at in here is the fact that if something was to be damaged on the tree, or if say for example, a piece of bark was damaged on the outside of the tree, although we're talking about apical and lateral meristems being at, say, the roots and the shoots, if that piece of bark is damaged, because the tree has such a complicated structure inside it, and the cells are so malleable in what they can do, meristems will come down to that location or develop in the location of the damage within the bark of the tree or within the cambium layer, and begin to grow and create kind of a healing patch or a scar within the tree. And I'm sure we've all seen scars on bark from things like this occurring. That's not just the cambium layer pushing out phloem cells. That kind of initial boost in growth to try and maintain the tree's health is coming from the meristem system. Okay, so what type of meristems have we already encountered in this module? So we've already started to talk a little bit about primary and secondary growth. So we're talking about lateral and apical meristems. Right, so we're looking here at tip growth. Alright, so we're trying to develop our understanding of that first question that I had on this slide about how our primary growth and how our secondary growth might occur. So, tip growth, meristem tissue at the apex or apex. Apical meristems give rise to primary plant body and are responsible for the extension of roots and shoots and the production of leaves and reproductive parts. Okay, so if we're looking here at the meristem cells consistently being pushed up, right? So if we look at meristems and we think about how these things are actually growing and how they take part in the tree, right? This is a meristem structure. So the way I want you to think about this is the very first bud coming on the top of a branch, okay? And then there's continual growth underneath from the same meristem structure, and the bud is getting bigger and it's getting pushed to the top. The very tip of that original bud hasn't changed, it's just being moved further up the tree, and then as it comes to this location or this graphic here, it's actually starting to bloom. So the bud is starting to open up and the first couple of leaves are starting to grow out of it. So again, the top of the bud is being pushed right out through, and it's started to actually develop phloem and xylem tissues to be able to transport minerals and water up and down to that new fresh growth. And then as it continues to grow, the cells from the meristem are pushed right up and continued on, and the trail of meristem here, the two yellow lines, will continue to grow up into the tree as the tree continues to grow, so that it allows for fresh buds to be produced in the future. Okay, and we've got our kind of differentiation between parts here when we start looking at phloem and xylem tissues, which is very important. Okay, so it's just important to recognize, and I find it easier when you're looking at this and thinking of an actual bud on a tree rather than a cell structure. So the bud is at the very tip of the branch, the small initiation bud, and then as it starts to grow again, it's getting pushed to the top. The tip of the bud hasn't changed, it's just been moved forward a bit more. Again, it's starting to open up here in a branch. The tip of the fresh leaf that was inside the bud hasn't been brought from somewhere completely differently. It was still at the very top of the branch or top of the bud, and has been starting to push its way out and start to open up for its first bit of photosynthesis, or and then continuing on here, that new fresh growth leaf or branch has started to create a good strong shoot coming out of the tree, and it's starting to get an actual structure to itself rather than just a leaf base. Okay, so this graphic on over here will give us the same understanding as to what we were just talking about. Okay, so on the right hand side here, we can see all our different tissues. We can see we're looking at a root system here, and we're talking about the different tissues within the root system. We can see the root hairs. At the very tip of them root hairs, we'll be looking at the likes of meristems, and then we're looking at meristems here, so apical meristems, coming down out from the root system and into the soil to help us get nutrients and increase the tree's ability to take minerals and water from the ground, also to increase the tree's anchor ability and be able to maintain its solid structure. Okay, so working our way back up the tree, then we're looking at a bark section or a trunk section of the tree, and we can see then this is a section we've seen before where we know what wood rays are. We know what wood rays are, medullary rays. We know that their function of them is to store starch. We've got our barks, cambium, phloem, xylem. So within that there, if an injury was to occur to the tree, our cambium layer can allow for meristems to actually help to repair the damaged location in the tree. And then if we work up to the very top, just what we were talking about there with the shoots, so we've got a cross section of a longitudinal section of a shoot system here at the top of a branch, and we're looking at young buds. We can see zones of large cells, and we can see zones of mature cells. So if we're looking at a mature cell structure, this could be last year's kind of final growth stage. We've got our fresh buds or young buds coming out through it. We've got large cell zones, zones of cell enlargement. That would be where the trees actually start to take its growth that year and is allowing for a lot of quick, rapid growth. So large cells would allow for large or quick transport of water and minerals to that location to allow for growth to take place very quickly. Okay. So primary growth process. So we've talked a little bit about this, and we've seen this image there a second ago. So division followed by division followed by differentiation. Okay, that's basically how this works. So we're dividing our cells, we're creating more cells as the meristem continues to create structures and create primary and secondary growth. And then when it gets to a certain point, the cell will then differentiate into its different aspects. So we've started off as a bud, we've pushed out as a bud and become a leaf, and then as our leaf has started to grow and crave food, it's actually grown its own branch and became an actual kind of part of the structure of the tree rather than just a piece of the tree that's going to create energy. Okay, and when it gets to the stage where it's become part of the structure of the tree, we have now got a branch, and within that branch we need to have our phloem and xylem cells to transport both minerals and water and food around the rest of the tree, as well as developing structure and strength within the tree. Okay, so we've got a couple of different types that it can differentiate into. So we've got dermal or skin cells, vascular or transport cells. So we're talking about xylem and phloem there. We've got ground or storage cells, so we could be talking about medullary rays there. We've got leaves, which is a primidori, and then we've bud, primidori themselves containing meristem. So buds are a very good example of where you can actually get meristem. And if you do happen to come across any cross sections underneath a microscope of a meristem, it's more likely going to be a cross section of a bud. Okay. So primary growth differentiation. So if we're looking at, we're trying to separate this down in between primary and secondary growth. Okay. So if we start at the bottom, so last 2 years we've got a lateral bud. Okay, so last 2 years of growth. So 2 years ago, this is where the growth had occurred originally. This was all taking place in this location 2 years ago. So 2 years ago, we're more likely going to see some lateral growth. It's very unlikely that we're going to get a lot of kind of fresh branches or fresh stems shooting out in this location. The odd time we'll get a cluster of buds, but usually they won't take off into any large branches. But we could and we will develop kind of small knots or small branches in lateral bud formations, allowing for more growth to take place, but this is not the primary area for growth. Okay, the year ahead of that then, okay, so the last year's growth, you're going to start to see a bit more activity within the tree. The bark will be fresher, it won't be as dark, there's going to be more evidence of buds taking place or growth trying to occur within that tree. And then if we're looking at a cross section of the tree itself, we can see all the different layers have developed very well within the tree. So it's actually differentiated quite well from the original cells. So we've got our vasculum cambium is created, secondary phloem is created, primary phloem. We've got our cortex, which is just like it's within the sapwood. We have our cork cambium layer, we have our cork, we have our primary xylem, and our secondary xylem cells. Okay, so primary are very much the early cells, secondary are the ones that are going to actually solidify and become the kind of main parts of the tree. Alright, now if we're looking at this year's growth, we've got a little bit less kind of development within the tree. So this is a younger section of the tree. We're talking a little bit more about initial growth stages here. So just like we were saying, when we're looking at the different cell structures within the meristem, so we're starting off very simple, and then as the years progress, we start to develop the structures within the tree that are necessary for growth and healthy structures. Okay, so we're looking back at the top here. So the initial bud tip is starting to grow. We've opened up this bud tip now in the cross section to have a look at it. We've got an epidermis, so we've created a very thin layer of bark basically on our epidermis or a dermal cell. We have our cortex, which is our structure or body of the tree, so eventually this will become sapwood and hardwood. We've got primary phloem, vascular cambium, so we've got vascular cambium, which is allowing for growth to take place as well, so within the cambium layer. And then we've got our primary xylem. So primary phloem, primary xylem, they're the initial parts of the xylem system and the phloem system, or the vascular system, that are going to allow for water to be transported up and down the tree, as well as any of the initial food produced, so glucose produced by photosynthesis, to be transported back down the tree. As that first year's growth continues on, this year will start to look like the second year there'll be more growth taking place above the tree as well. So we're looking at our primary here, we're going vertically up through it. Okay, so summary of primary development. So apical meristems, then we can divide off into three main areas. We can look at proderm, so epidermis, which is where eventually going to become part of our bark or underneath the cambium layer. We've got our ground meristems, so we're looking at cortex, pit rays, so that's the structure within the tree itself. And then we've got the procambium, so the procambium is going to become the cambium layers, and it's going to become our phloem and xylem system, so it's basically our vascular system within the tree. Okay, so secondary growth. We've already seen these graphics before, and we should have an understanding of what's going on here. So this is something that should almost be coming up on your applications principle 2. Okay, so we've covered this in detail in the past, but now you have an understanding of it and how relevant it is to primary and secondary growth. So you understand how primary growth works now. We have looked a little bit about how the tree grows itself before, and now we're putting a name on this as secondary growth. So it's important that we just pay attention to what's happening here. Okay, so we know as well we've got our cambium layer, we've got our what we call our inner bark or our bast, we've got our sapwood to the right. But what's important to us that we understand that towards the bark from our cambium layer we have our phloem tissues. Towards the center of the tree from our cambium layer we have our xylem tissues. Our xylem tissue will eventually become sapwood, and then as it matures and becomes older, it'll eventually become hardwood. We've got our xylem tissues or our inner bark, okay, which will eventually become our outer bark. Our outer bark is just dead xylem tissues that have been, or phloem tissues that have been pushed to the outside of the tree and have become kind of hard and rigid and used to protect the growth structures of the tree and the transport system within the vascular area. Okay, so similar to what's going on in the meristems at the top of the tree. So for our primary growth, we're looking here at our secondary growth, and we know that if we're looking at this, we're looking at our kind of light green cell here, which is our vascular cambium layer. So we're adding phloem to the outside, we're adding xylem to the inside. We're going on then and we're adding on more phloem, more xylem. We're looking at basts, and it's continuing on and adding to the outside, adding to the inside. And our xylem cell our cambium cell more or less isn't changing its locality as far as how many cells are within the new place, okay, or within the area. So it's just important to understand this and see how it works, okay. So a little bit more detail on our secondary growth. So our lateral meristems, we're looking at the vascular cambium ideas. Okay, so we said that there. We're just looking at that. Our cambium divides radially. So what's the best way to explain this? Yeah, so it divides radially. It doesn't divide um it doesn't divide vertically or anything like that. It divides radially, so we're able to add on annual. The best way to understand this and see what that means, by dividing radially, we're talking about annual rings. You know, so we're adding on more concentric circles as we're going. So we're talking about the radius of it. Okay, so as that radius increases, we also need to increase the circumference of that circle, and that's where we start to look at cambium layers dividing tangentially. To allow for that increase in diameter or circumference, we need to insert more cells between the gaps, basically. So this helps to create ray cells. Okay, so we've got periclinal divisions and then anticlinal divisions. Okay, so basically radial and tangential divisions, allowing for the increase in diameter and allowing for the increase in circumference to fill the gaps. Okay, so as you can see, in an anticlinical division, okay, we start to get this almost radial line. If we were to draw a line through the center of these two cells towards the radius, okay, or towards the center of the tree, this allows for the development of ray cells. Okay, so we can create a parenchymal ray coming through this area here, which is creating a channel that goes straight through the tree, allowing for storage of starch and also allowing for the transport of minerals and foods within the tree to allow for healthy woody fibers within the tree itself. Okay, whereas if we're looking at a standard kind of radial development or parenchymal development, we can see that there's almost walls created to prevent that. So there's very little transport occurring in between these two cells towards the center of the tree, in comparison to allowing for a ray cell to come between the two cells in an anticlinical division. Okay, so Fusiform, initial general purpose of cells. Okay, so fusiforms are, it's interesting enough. So if we're talking about cell structure and general cell structure itself, okay, so if we're looking at trachoids, we've had a look at trachoids before. We've talked about how trachoids originate and then they develop into vascular cells, they develop into structural cells. And what we're looking at here is our fusiform cell. So what we're talking about is how a cell actually starts. So this could be within our cambium layer. As our cambium layer is beginning to grow and we've got our secondary growth, these cells need to change slightly. So if we're looking at the say radial growth, we're going to have maybe development of trachoids, we're going to have the development of vessels, we're going to have the development of kind of vertical kind of cell structures to allow for transport of minerals or food up between the tree. Whereas we know that during the increase in diameter of the tree, we also have to increase the circumference, and we need to fill them gaps within the cells. And that's where a fusiform comes in, where we're able to actually change from trachoids, parenchyma cells, and we're now looking at the development of ray cells. So Moderna ray cells, or medullary ray cells, where the cell is now actually almost going to be horizontal and increase within the gap between the already existing vessels and trachoids. Okay, allowing for storage of starch and allow for transported material towards the center of the tree. Alright, and here we're looking at just say insert of parenchymal model, and we're just looking at the parenchymal model of divisions of cambium fusiform initially. So more or less this is just a more complicated version of what's going on here. In this image we can actually see it occurring at a more kind of in-depth structure. Okay, so we're starting off as our one cell. As we're starting to grow in diameter, the cell is starting to divide. To allow for that division, we get a radial increase. Our radial increases, our radius continues to increase, and then we need to produce more cells to allow for the increase in diameter. So we start to create cells that are kind of rotated in an opposite direction, and when them cells are created, initially a ray will begin to start, and that's going to allow for the storage of starch and transport material towards the center of the tree. Okay, so cells. Not going to read through this too much. You can have a quick look at it and try and make your own understanding from it. We have talked about all of these cells at this point, and this is a nice little image. It's giving you the location of it, it's giving you the function of everything. Okay, so trachoids, mainly softwoods, some hardwoods, provide strength and structure. We know that. Parenchyma cells, we're talking about softwoods and hardwoods. We can get them anywhere. They conduct and store food. Fibers found in hardwoods. Vessels and pores found in hardwoods. One provides strength and one conducts sap. Right, so now we're looking a little bit more about softwood hardwood distinctions. Okay, so softwood hardwood distinction. So straight away from this, you should be able to think back to even your junior cycle understanding and talk about what we see in softwoods, what we see in hardwoods, what distinguishes the two. Are we talking about structures? Are we talking about seeds? Are we talking about bark types? Are we talking about conical shapes? What are we talking about? Evergreen? What comes to your mind when we start to talk about distinction between softwood and hardwoods? So softwood gymnosperm. So gymnosperm seeds are usually formed in unisexual cones known as strobili, exposed on modified leaves, and the plants lack fruit and flowers. Okay. Hardwoods are angiosperms. Seeds of angiosperms develop in ovaries or flowers and are surrounded by a protective fruit. Flowers can be unisexual, example male flowers and female flowers, or bisexual, the flower has both male and female parts. Okay, so we're looking at angiosperms, we're looking at gymnosperms. So if we take a quick look at the angiosperm structure, we have a kind of a pine cone, as would be described, or a cone, and we have a closed cone. Within that cone, we can see the different pieces of seeds stored within the cone. We have our cone scale here, so this is looking at one scale of the tree. So we've got immature seeds basically sitting flat on each one of these cones. Okay, as the cone begins to open, the ripened seeds will blow out of the tree or fall out of the branch, and it's ready to go. It doesn't necessarily have to fall off the tree. However, when the cone does fall, it can create an impact, and the seeds can be sent flying in different locations or different directions. And then when we take a look at the angiosperm seeds, so we're talking about fruits really. So if we even look at a tomato or we look at an apple, so more so towards an apple here, it's very easy for you to understand. So our seeds are in the center of the tree. We've got fruity flesh on the outside. That flesh is creating basically a red flag to animals in the area. We might be trying to disperse these seeds through animal distribution. It's also creating initial food storage for the seeds. If that apple was to fall on the ground and start to decompose, it's actually helping to create an environment that will initiate growth for the seeds itself. Okay, so we can see the difference between the two. So if we look even more simply, we're talking about Chinese lantern here as a style of a seed, but if you think about a horse chestnut tree, it's all very similar in structures. But angiosperms, gymnosperms are different, and it's important to recognize that, and it's very easy to kind of forget which is which. But this is something that's going to be coming up and we talk about all the time, so make sure you're paying attention to that. Okay, so what we're going to have a look at, I'm going to talk about it a little bit more in Friday's lecture, okay, is your case study on silviculture, which is due in week 9. Okay, so we're currently on week 6, we're giving you week 7, 8, 9. We're going to have a bit of lab time allowed for this now next week as well. So your case study on silviculture resource. So students will research the topic of silviculture in Ireland and design a resource Students will research the topic of silviculture in Ireland and design a resource to aid in the education of the public based on this topic. Students will need to express knowledge gained through lectures, labs, and additional research. We may ask for a case study of a selected area local to the student. The resource could be a poster, website, etc. The media selected should be rationalized prior to completion of your work. So basically, I don't want to have a rationalization. You don't hand me in a letter or send me an email saying I'm doing it this way because. But if we were asking you why you done it with a certain media or why you done it with a certain source, you should be able to explain your points for it. Okay, so pretty much we've kind of already started these topics and we've already kind of helped work towards this with our last 2 applications of principles. Okay, so we've done a little bit about silviculture. You've already been designing posters for schools. You've already been kind of designing an application to prove why you are going to plant the forest in a certain way. So this silviculture in Ireland case study isn't something that's going to be completely out of the blue. You'll have already been looking at topics similar to this, and within this area now all you have to do is design something very simple to explain to the public what silviculture is. Okay, so even the application principles that are due next week, we're talking about how a tree grows. Before that, we talked about why we plant forests in certain ways. Just be careful when you're doing this that you don't mix up silviculture and forestry. they're two different things. Okay, so I'm going to talk a little bit more about this on Friday, but try and just realize that this isn't going to be the end of the world. It's something that we've already started looking at and we've started moving towards through our applications of principles. So I wouldn't be worried about it in any way. All right. So other than that, thank you very much. And as always, don't be afraid to send me an email or ask me any questions that's going on. Get in contact with Maria or Edmund if you have any issues, and I'm sure we can work towards that. Okay, so I'll spend a bit more time on this case study now in tomorrow's lecture. Okay, so thank you very much.